Insulin's pretty interesting. The receptor is dimeric, which means they're the two parts of they're two polypeptides, and they're mirror images of each other. And so they're floating around in the membrane, and when insulin comes in, they come together and activate. And it's a tyrosine kinase. And so it's a really weird receptor in that once insulin brings the two pieces together, and it forms this ladder here, on the intracellular side, that tyrosine kinase is activated and it phosphorylates itself and causes all of the effects of insulin. Now, what's interesting is that typically when you have two hormones that are antagonistic like this, right, which means they simply do opposite things, like insulin causes an increase or decrease in blood sugar, right, and glucagon causes an increase in blood sugar. They act on the same tissues, which means they have to have different second messengers, right? So that's important. And so insulin has a bunch of weird, new kind of setups here with IRS1 and this phosphoinositol 3 kinase, which is kind of like phospholipase C, but it's a kinase, not an enzyme, and it phosphorylates your inositol, so it stays in the membrane. That's really the only difference between it and phospholipase C. Instead of releasing the triphosphate, the IP3, we just keep it in the membrane, and that complex allows you to activate protein kinase B, and that then does all the work of insulin. So, whereas glucagon activates adenylate cyclase, right? Like I talked about in the video. So. But since you've already heard it, I won't go over it again. I bore you. So, all right. So let's start talking about the hormones. At least I want to talk about, right? So, again, I'm trying to be theme-based here and make sure that you all kind of understand how the endocrine system works. I'm not going to make you memorize every hormone there is, right? But the classical endocrine organ here is what's typically referred to in a physiology text as the master gland, is the pituitary gland, which is this little outgrowth of the floor of the brain here, the floor of the hypothalamus really, that releases a number of hormones that control a lot of the endocrine glands of the body. And what this picture is just out of a human anatomy text that shows you all of the endocrine organs, or most of them at least, that are found in the human body, which means all of these organs release hormones, right? Now, the one thing I do like to touch on, outside of the classical endocrine organs, there are a number of paracrine molecules that are very localized, that are very important in the physiology of cells, particularly immune and inflammatory responses, are controlled by a whole bunch of paracrines. And what a paracrine is a communication ligand that acts locally, right? Wonderful example, histamine. Right, histamine is a wonderful example of a paracrine ligand that is released just locally to cause inflammation, and is controlled. The release of it is controlled by a cell that's called a mast cell, and it just releases histamine in a very local area, and the inflammation only happens there. But there are lots of molecules called cytokines, like interleukins, maybe you've heard of before. These are molecules that are released by the immune system, by immune cells, or by tissues that cause an immune response that are very localized. And if you just look up all the cytokines in a body, there's a poster somewhere that I once saw that showed all the cytokines together, and it is overwhelming, right? So immunologists are incredibly smart people, like Dr. Stemke, smart dude, man. He's an immunologist, and it studies all those cytokines and how they work. But we're going to stick to the endocrine system. So remember, you have two types of glands in the body. You have exocrine glands, which is this one right here, and you have endocrine glands. The big difference is an exocrine gland produces a product, so does an endocrine gland, but the exocrine gland secretes it into a duct, and that duct drains somewhere, right? So it drains that product into a body cavity or onto the surface of something. So skin glands are exocrine glands. The digestive glands, salivary glands are exocrine glands for the most part, dumping their products into ducts and then releasing those products into a body cavity or onto the surface. An endocrine gland produces its product and just dumps it into the interstitial space, no duct involved, and then it travels into the blood and then is transported somewhere to its target tissue, which has the receptor. And it's long distance traveling typically, right? As we've mentioned in past lectures. Now, the pituitary is a great example of a number of really important hormones. So the pituitary hormones are really the ones that I focus on, and there are a number of them. And then we'll talk a lot about how thyroid hormone works, all right, so that you can get a really good example, right? Now, I used to do a lab with tadpoles. It's a wonderful lab, but it is not nice to the tadpoles. And so, you know, over the years, students just, they get too upset to do it because it kills the tadpoles and they change really fast, and they can't keep up. And so it's you know, it's overwhelming for some students. So I don't do it anymore because, you know, killing tadpoles isn't all that fun. That's why we do the ADH experiment instead, which I've done. I used to do them in tandem because, you know, we don't kill the frogs, right? The frogs are happy. They're up there jumping around. They're happy. So
they're fat. They eat so much.
Every once in a while with that experiment, a frog will react. Some frogs are sensitive to ADH. It can cause massive vasoconstriction in some instances. And every once in a while you have a frog that is already stressed that has trouble with it. But typically they're absolutely fine. So we don't do the tadpole experiment, but it is an incredible experiment. So thyroid hormone, and I'll talk about it later, controls metamorphosis in tadpoles, and we'll talk about what it does in a little while. But thyroid hormone is also very unique as far as the way it's produced. And so that's why I kind of chose it as our model hormone. Plus, there's tons of thyroid problems in the human population, right? So a lot of people have hypothyroidism or hypothyroidism. It's genetic, and it can affect them dramatically. So let's talk about the pituitary a little bit. So the pituitary has two lobes. So it's basically a ball of tissue that's sticking off of the bottom of the hypothalamus, or what we call the floor of the hypothalamus. So basically, part of the pituitary is just an outgrowth of the hypothalamus during development. The nerves just grow out and form a ball of tissue that's called the posterior pituitary, or it has a big long name. Hypophysis, as you'll see in a minute, means pituitary. If you put neuro in front of it, then it's the nervous pituitary, right? Is the basically posterior pituitary, which is an outgrowth of brain tissue or of the nervous system. The anterior pituitary, which is stuck to the surface or to the side of the posterior pituitary, is a epithelial origin ball of cells that came from the roof of the mouth during development, or from the oral epithelium, and is not directly attached to brain tissue, all right, or isn't an outgrowth of the brain. And it produces a number of hormones. So these are hormones all on the bottom here, are anterior pituitary hormones. And you'll see a name called adenohypophysis. Hypophysis means pituitary, adeno means to secrete. This has to do with its epithelial origin. So the posterior pituitary, can't speak. The posterior pituitary is nervous tissue. The anterior pituitary is epithelial tissue. So these are the anterior pituitary hormones. So you have 1, 2, 3, 4, 5. There is a fifth that you'll see on one slide, but I won't talk about it or test you over it. It's called MSH. I'll show you it in a second. But these are the major ones that you find throughout vertebrates that all have major implications physiologically in homeostasis within all vertebrates. We'll go through these here in a second. And you're going to see a lot of mammal, physiologic responses. But we already talked about how important prolactin is in fish gills, right? Remember when moving from salt to freshwater, prolactin was one of the hormones that allowed that change to happen. So now, when you look at the anterior pituitary, what I want you to notice is a specialized setup of blood vessels here that will remind you of the blood supply to the glomerulus when we talked about the glomerulus. And this is a setup where you have two capillaries beds in tandem that are connected by a blood vessel that's called a portal blood vessel. So portal vessels connect capillary beds in tandem. Typically, that's not the normal setup in the circulatory system. You remember, capillaries are the smallest blood vessels, and that's where we get exchange. And notice we have two capillary beds right next to each other that are hooked by these two portal vessels that are called pituitary portal vessels. And these support each other. So the problem is the hypothalamus, in reality, is the major controller of all of the pituitary gland. Now, with the posterior pituitary, it has direct control of it because the nerves originate in the hypothalamus and their axons travel down into the posterior pituitary. So when those nerves decide to fire, they release their products into the blood, right, directly. But the hypothalamus doesn't have a direct connection to the anterior pituitary because, remember, it came from somewhere else. It has a different origin. So I have to control it using hypothalamic hormones. To do that, I need nerves up here to be able to release their product into the blood that is directly sent to the anterior pituitary. And that's why you have this portal system here where you got two capillary beds in tandem. The first capillary bed sits over the top of the stalk here, or the outgrowth of the pituitary. And there are hypothalamic nerves that are really short, that travel right to the edge of the capillaries, and they release their products into that first capillary bed. And then they travel through the portal vessels and leave the second capillary bed to control the anterior pituitary cells. And these are called releasing and inhibiting hormones. So they'll be called hypothalamic releasing and inhibiting hormones that are going to cause either the inhibition or the release of the hormones from the endocrine cells that make up the anterior pituitary. So here's a picture of the posterior pituitary, and this is kind of a drawing from another textbook that shows you the posterior pituitary is nervous tissue. Remember, nerves, I kind of drew a picture of one once. We're going to get to the nervous system. Has a cell body that has a bunch of little processes that come off of it called dendrites, and then it usually sends one long process out, right? A long wire, if you will. That's called the axon. So the cell bodies all originate up here in the hypothalamus, and they send their axons through the stalk all the way down to end in the posterior pituitary. And they simply release their product into the interstitial fluid, and then that will enter the bloodstream directly. And so when you look at the posterior pituitary, what you're going to see are under a light microscope, a bunch of striations or stripes that are the axons. When you look at the end of the axons, I know this is hard to see because it's a very light image. These are the ends of the axons here, and this is the wall of a capillary, and this is the lumen of the capillary, and these little dark circles are vesicles that hold the neurohormones, and then they release those neurohormones into the blood supply. And one of them, because we played with it yesterday, which is ADH, all right, or what's called vasopressin. That's one of the posterior pituitary hormones. Is ADH, antidiuretic hormone. The other one is vasotocin. which it causes smooth muscle contraction. There are only two hormones that the posterior pituitary produces, ADH and vasotocin. All the rest of the pituitary hormones come from the anterior pituitary. So the adenohypophysis is the anterior pituitary. Now again, this is a wonderful picture up here of a pituitary from a rodent brain. And what I love about it is this is the third ventricle right here. So do you guys know what ventricles are in the brain? They're just big fluid-filled cavities. Your brain is hollow. Mine's more hollow than most. That was a joke. Anyway, all right. So there are these holes or spaces, cavities, large cavities that are called ventricles. And so this is called the third ventricle. All right, and the floor of the third ventricle is brain tissue that's called the hypothalamus. Now, the hypothalamus is an interesting part of the brain. It is in control of a lot of homeostasis, right? So that's its job. It has a bunch of clusters of nerves that are controlling specific parts of homeostasis. One of those, or a couple of those clusters, control the pituitary glands. So what's cool about this is notice, what I love about this picture is that notice this is part of the brain up here, right? And it's kind of a light color. And notice the posterior pituitary is the same color, isn't it? So you can see it's because this is nervous tissue, right? The axons, they travel from the hypothalamus down into the posterior pituitary. Hundreds or thousands of them do. Whereas the anterior pituitary stains completely different. It's darker, right? Because it's not nervous tissue, it's epithelial tissue. And it sits within the skull, within a depression in the skull here called the silica turcica, which is this little depression right here. And this is all bone tissue here, histologically. This is a nicely done slide, as they had to decalcify the bone to get that section, right? And this is a nice enlargement of the anterior pituitary. So adeno means secretory, hypophysis means pituitary, so this is anterior pituitary. And these are kind of the regions typically in a mammal, if you're looking at the front of the anterior pituitary, and what hormone regions you have here. So these are hormones, GH, ACTH, and TSH. And then there are some small areas here that are in red that are going to release what are going to be called FSH and LH, which are responsible for reproduction. And so we're going to talk about these different hormones that come from the anterior pituitary. Now, the cells of the anterior pituitary are called troph cells. All right, they're called trophs. And notice there are 5 of them. 5 different types of trophs or cell types, and they're called thyro, gonado, cortico, lacto, and somatotrophs. Each one of them produce different hormones. They all look exactly alike. The only way you can tell the difference between them is to stain them using immunocytochemistry, right, where you actually stain the hormone so you can see where the cells are found. And that's how they came up with this little map, right? Because these cells on the edges would stain for growth hormone. The ones in the middle here would stain for ACTH. And then this little island in the middle would be TSH, right? And so you can see those, that's what these pictures represent, are immunocytochemistry labeling of individual types of hormones. So these guys that are in brown up here are what are called thyrotrophs because they are staining a hormone called TSH, thyroid stimulating hormone, or it's called thyrotropin, all right, as you're going to see. Gonadotrophs, they release gonadotropins, all right? And so when you hear that word tropin, all right, when you hear the word tropin, a tropin is a hormone that causes the release of another hormone. So tropin causes the release of other hormones. So Thyrotropin, TSH, is going to cause the release of thyroid hormone from the thyroid gland. Gonadotropins are going to cause the release of the sex steroids, those hormones from either ovaries or testes. Corticotrophs, they produce a tropin, ACTH, which is going to control the adrenal glands. And then lactotrophs, they produce prolactin, all right, which is, they're called lactotrophs because they were first discovered in mammals. So is prolactin, even though it's found in other vertebrate groups. You stick with the original name. And prolactin is going to control milk production in mammals, right? And hence where this name comes from. So it works on the mammary glands. And then somatotrophs, they produce somatotropin, which is called growth hormone. All right, and growth hormone controls hormone release from the liver. So this is a list of them for you, right? So that you have them all. So here are your 5 cell types, and here are the hormones that are basically produced. Now notice this one down here I haven't mentioned, right? So here's the MSH, melanocyte stimulating hormone. In vertebrates, it typically causes pigmentation in the skin, all right? So MSH causes pigmentation in the skin. It doesn't in every vertebrate, but it does in many of them. But what I want you to notice is that all of these hormones in this list are tropins, except prolactin and MSH, right? And since we're just going to ignore MSH, it's just prolactin for you guys, right? So prolactin is the only one that's not a tropin. It's the only one that doesn't control another endocrine gland. This is why the pituitary, particularly the anterior pituitary, is called the master gland, because it releases mostly tropins, doesn't it, that control other endocrine glands. And so you can see over here, this gives you kind of the tissues or the target tissues. So the one thing that's not listed up here is that human growth hormone. It causes the liver to release insulin-like growth factors. ILGs, insulin-like growth factors. So there's IG1, there's ILG1, or there's IG1, sometimes they're just IGs. So insulin-like growth factors are hormones that cause, for example, body growth. They cause, they help cause mitosis at the growth plates, for example, during adolescence, along with the sex hormones. They cause growth. So they're insulin-like because they look a lot like insulin, like the hormone insulin. And they can cause increases in blood glucose levels. So a lot of times with pregnancy, women will have either they become insulin tolerant or they have diabetes, right? Gestational diabetes can be caused by the release of these during pregnancy, which causes the baby to get really big, but it also causes them to become very sensitive to insulin. So. And can cause high blood sugar. So these are the hormones, these are the targets. All of them are tropins except for prolactin. Okay? Here's another picture that shows you kind of all the troph cells, what they release. Notice it shows you all the tropins here. So here's thyrotropin. Here's adenocorticotropin, or ACTH. Growth hormone. It's also called somatotropin. That's not on here. Gonadotropins. Here are your 2 gonadotropins from the gonadotrophs. LH is called luteinizing hormone, and FSH is called follicle stimulating hormone, which is on the previous slide. They control ovaries and testes. They control secondary sex characteristics, the release of estrogens, the release of testosterone, the production of eggs and sperm. So they're controlling reproduction, right? The gonadotropins. I like this one up here. This is why I chose this one, because it does give you the mammal function of prolactin, the growth of breast tissue, the production of milk, right, from the mammary glands. But that's only in mammals, because obviously other vertebrates don't have mammary glands, right? So it helps with reproduction, water balance, and caring for young. So it causes a lot of behavioral characteristics of maternal instinct and that kind of stuff. All right, so one other thing I like to talk about are classes of hormones. Right, there are a whole bunch of different ways to kind of organize the endocrine system, but here are kind of the three major groups of hormones based on their chemical class. So you basically have proteaceous or protein-based hormones that we call peptides or proteins. So peptide, what's the difference really? Because isn't a protein a peptide, a polypeptide? I mean, it's really, pretty much the same thing. A peptide is typically just a short protein, where a protein is big, right? But they're all proteins in reality. Whenever you put two amino acids together, right, it's basically a protein. We have steroids, which we kind of showed some pictures of. And then we have what are called amine hormones. Often they're called biogenic amines, because they're an amino acid that's been changed a little bit structurally, and then they become very biologically active or physiologically active. And that's the ones I like to focus on. We already talked about insulin. Insulin is a protein, right? It's a polypeptide. We talked a little bit about steroids, like the sex steroids. They're cholesterol based. Now let's talk about some amine hormones. So amine hormones are basically made up of amino acids. And here are some examples of the amine hormones. The first two up here are thyroid hormones. And that's what they look like. Don't have to draw them, right? But just to show you pictures of them. And then here are your three biogenic amines: norepinephrine, epinephrine, and dopamine. And man, when you look at them, they look really similar, don't they? They're all made from tyrosine. All of these are. All right, tyrosine is an amino acid. And the one thing that tyrosine has is this benzene ring, right? As you can see. So notice, it's the major constituent, or R group, of all of those hormones. Why benzene? Because it's got great three-dimensional shape, right? So it'll fit a receptor really nicely. It's also very stable, right? It's very stable and hard to break down. So it's got long-lasting effects. And really, all that we do with these three hormones down here to cause dramatic differences between them is change the groups attached to the amine end here. So notice what we've done with epinephrine and dopamine is we simply, have changed this group, right, here a little bit. So we've added a methyl group for one of our hydrogens. And the norepinephrine looks a lot like epinephrine, right? But we don't have the methyl group attached to it. And so we also have two hydroxyl groups here on that first carbon coming off the ring that dopamine doesn't have. And you can imagine these can be made all in the same metabolic pathway. They are, right? If I want to make dopamine, I have to make epinephrine first, and vice versa if I want to recycle either of these. So the hormones the enzymes that control the production of these hormones are in the same metabolic pathway. Now notice the thyroid hormones up here. The only difference between the two is how many iodine atoms are attached to it, right? You have 4 on the top and 3 on the bottom. So the one on top is called T4, 4 iodines, and the one on the bottom is called T3, 3 iodines. Both of them are thyroid hormone that can cause the physiologic response that thyroid hormone brings about. So we're going to talk about these two in particular and how they're produced later in the lecture. Now, proteins, as we've talked about before, are going to be produced via the endomembrane system, right? And so this just again shows you how this works. And insulin, for example, is produced as what they call a pre-pro hormone, right? Pre-pro hormone. So what the heck does that mean? That means that the messenger RNA produces a polypeptide that has a number of amino acids that are removed from it, both pre and during transcription, I should say. I'm sorry, during translation, I should say, or post-translational afterwards in the ER or the Golgi apparatus. And so the pre-pro hormone is the polypeptide that comes off, the entire polypeptide, that has a little sequence on its edge, or the first, let's say, 8 or 10 amino acids are called the signal sequence. All right, and that's what that P stands for right there, all right, is the signal sequence. And that signal sequence, what it tells the cell to do is to attach the ribosome to a shuttle protein, all right, that will carry it to the rough ER. And so those first like 10 amino acids that are translated in the free ribosome out in the cytoplasm are the signal sequence that the shuttle protein recognizes. It grabs onto that ribosome and then shuttles it to the rough ER. And in the rough ER, that signal sequence is immediately cleaved off because it's useless once you're producing the protein. And so that's where the pre-pro hormone then becomes the pro hormone, and that happens immediately in the lumen of the ER. You get rid of the signal sequence. Notice with insulin, we have a bunch of cysteine amino acids, because how do I know that? Because there's lots of sulfur groups. See all those sulfur groups? So that is the R group of cysteine, which is a sulfur-containing amino acid. And those will allow this protein to fold over itself and form disulfide bonds, which happen in the Golgi apparatus as it is processed and matures. And so you can see you take your polypeptide and you form these disulfide bonds that give it very stable three-dimensional shape. And what you can notice here is the proinsulin is actually two pieces. It's the C peptide, what they call the C protein, and insulin. And so there's an enzyme that cuts them or cleaves them inside of the vesicle right before secretion, and you get your C protein here, or your C peptide, and then your insulin molecule, which notice has nice three-dimensional shape. It's nice and flat, and it fits really nicely into that dimeric insulin receptor. That's released into the bloodstream, and that's what travels through the body and causes a systemic effect, right? Insulin is a systemic hormone. It causes all body cells to recognize sugars, right? By causing GLUT transporters to be inserted into the cell membrane so you can remove glucose from the blood supply. And that's how it lowers blood glucose levels, right? In the body. And this is cool. Depolarization of the cell membrane triggers exocytosis. You know that mechanism, right? You learned it a long time ago. Remember that? Beta cell release is a depolarization because of closing of ATP special potassium leakage channels. Remember that? Which causes calcium voltage gated channels to open. So depolarization causes those calcium channels to open, which then causes the vesicles to release insulin. The only thing I'm going to do about steroids hormones is show you them. Now, steroid hormones, the one difference between proteins and amino acids is you can produce them and store them in vesicles, right? Because they're hydrophilic. They're water. They dissolve in water, right? Or at least part of them does. And so you can store them in vesicles. Steroids, you can't do that. You can't make them early and store them because cholesterol is hydrophobic. If you are just making them continuously, you'll just continuously release them because they just move across the membrane by diffusion. So you have the steroid-producing cells have big droplets of cholesterol inside the cytoplasm that are kind of their resource, right, that they use. And when they're stimulated, they make the steroid real time. And then they release the steroids into the bloodstream. These have to be carried by transport proteins. all right, because they're hydrophobic. And here are some of the more common steroids: cortisol, aldosterone, testosterone, estradiol. Look at these two again. My gosh, isn't it amazing? Those hormones look really similar, but boy, do they cause very dramatic differences. I've been married 25 years. I have no idea what my wife's thinking most days, right? And she produces a lot of this, and I produce that, right? We call it the dumb hormone, don't we? Testosterone. Females produce testosterone and convert it to estrogen. Males do the same thing. We can convert testosterone to estrogen. We just don't have a lot of the enzyme required to do that. Females do, obviously. That enzyme, by the way, that converts testosterone to estrogen is called aromatase. So it's this enzyme right here. So aromatase takes all androgens. You can see, I say androgens because they're male kind of hormones like androstenedione and then testosterone or DHT. It can take all of those and produce estradiols or estrone. Estradiol is the potent form of estrogen produced by the ovaries. Males produce estrogens in the brain and in the male reproductive tract mostly, in parts of it, but in very low concentrations. The other place testosterone is produced, just for your information, I won't test you on it, is the adrenal glands produce testosterone too. So that's where females produce their testosterone that's released into the blood, and it drives like sex drive, part of the reproductive cycle. And that testosterone comes from the adrenal cortex, actually, in females. So, I always wanted to teach a vertebrate reproduction class. Wouldn't that be a cool class to teach? I just don't think anybody would take it, unfortunately. All right, so far so good? All right, so hormones, where their receptor is depends on what type of hormone it is, right? What class it is, whether it's hydrophilic or hydrophobic. So hydrophobic hormones like steroids or cyclic compounds or eicosanoids, these again are kind of cytokines, right? Their receptors can be internalized because they can pass through the cytoplasm. They can go, their receptor can be found in the nucleus, a matter of fact, because these things like steroids can pass right through all the cell membranes and find their receptors. And often a hydrophobic hormone will turn on gene expression directly. So what that means is it'll go into the nucleus, go to a gene, and cause transcription, right, of that gene. Hydrophilic hormones, like amines and small proteins, their receptors have to be found on the surface because they can't pass through a cell membrane, right? And so they're going to have to cause the activation of second messengers, right? And we also know these guys happen to be typically really potent because you don't need a whole bunch of them to reach their receptor at the surface to cause major effects because of amplification. Because they all activate kinases, don't they? That cause kinase cascades. So nuclear receptors or cytoplasmic receptors for hydrophobic hormones typically lead to direct gene activation or gene expression. Membrane-bound receptors are going to activate second messengers like cyclic AMP, cyclic GMP, IP3, right? Calcium, all of those second messengers we talked about. Here's a really good example of how a steroid works. So steroids, as I mentioned, are carried through the bloodstream using a carrier. They're dumped at their target tissue. Now, their target tissue typically will have either a cytoplasmic or a nuclear receptor. This receptor really is a shuttle, is what it is. And that shuttle will carry it into the nucleus to a specific region just above the promoter of a gene. And it looks like this, all right? So this is the setup of a typical vertebrate gene, right? And if you don't remember what a promoter is the area of DNA that is recognized by the enzyme polymerase, right? So polymerase can bind to that when it's exposed. Often the way we think it works, and believe me, I'm not a molecular biologist, right? Is that often the promoter is bound up, right? Or it's twisted so that the enzyme can't get to it. So we have to relax the DNA in order for the polymerase to find the promoter. And that's where the hormone comes in. The hormone is going to bind to its receptor, and that receptor is going to shuttle it in to this gene of interest. And what it's going to bind to, the hormone and its shuttle, it's going to bind to a sequence of DNA upstream, just upstream of the promoter, called a HRE, all right, which is a hormone receptor element. That's what HRE stands for, a hormone receptor element, which is the sequence of DNA the hormone and its receptor recognize. When they bind to that, that will shift or open up the promoter so that polymerase can see it, right, or bind to it. And once polymerase binds to it, then we can start transcription, right, and produce messenger RNAs. So basically what we do is we open up a gene so that we can transcribe it, get many, many copies of the messenger RNA of interest, which can then leave the nucleus and be translated at the ribosome, and that causes what we call gene expression, right? So it's direct activation and protein production here through gene expression. Now, you can imagine that takes a little while, doesn't it? The hormone's got to travel there, it's got to enter the cell, bind the receptor, enter the nucleus, open up the DNA, transcription starts, messenger RNAs have to leave, you start producing proteins, so protein's got to build up a little bit, and then all of a sudden you get this physiologic response. That can take hours to days, right? Just depending on what process we're talking about. So that's what we call the slow response, all right, of a hormone. Now, some hormones cause an immediate response. If they're going to cause an immediate response, when I say this, some steroids that are hydrophobic can cause an immediate response. Well, they can't do it this way then, right? That takes time. So there are some steroids that actually do have surface receptors that they bind to also, and they cause a rapid, immediate response. And how do they do that? Well, like any hydrophilic hormone, they cause the immediate production and activation of a second messenger that brings about a kinase cascade that happens immediately. So they could do that first and then slowly turn on gene expression. So they can do both. There are steroids that do this, like cortisol does this, for example. It can bring about slow, more permanent stress changes and immediate responses to stress. Like, oh shit, I got a test tomorrow, right? And it's immediate. And so it can bind to two different types of receptors. Pretty cool, right? How that works. So I love this chart. So this chart's out of the Animal Phys textbook, actually, that shows peptide hormones here, like insulin, right? Steroid hormones, like we just kind of talked about. And then these are the ones we're going to end with, which are the amine hormones or tyrosine derivatives, which include two groups: catecholamines, or what I call biogenic amines, and thyroid hormone. This word here, catechol means benzene. So it's a benzene amine. So you take a benzene ring and you add an amino group to it. I mean, that's what you do. All you have to do is look at the structure of epinephrine, dopamine, or norepinephrine, and you'll see a benzene ring and then a modified amino group, right, or amine group. And that's where the names come from. I call them biogenic amines because once you put them together like that, they have very potent physiologic responses. We are going to kind of talk about thyroid hormone mostly as far as how we produce it and its importance. Now that's the last slide we'll end on here. So now, interestingly, hormones can bind to different types of receptors. right? That is, some hormones have 2 receptors instead of just 1. All right, and so epinephrine, for example, has 2 types of receptors, an alpha receptor and a beta receptor. And as a matter of fact, epinephrine and norepinephrine bind differently to them. Now, do I care about that? No, I don't. I just want you to, for the test, I want you to realize that, epinephrine binds to the alpha receptor better than norepinephrine, and norepinephrine binds better to the beta receptor than the alpha. And so they can have very dramatic, different effects at different tissues. But some target cells produce both receptors. So when they produce both receptors, the same target, that means epinephrine is going to bind to both, right? Whether it binds to one better than the other, it doesn't matter. It's going to bind to both, all right? And so you can get 2 different kind of flavors to that. It adds kind of a richness of stimulation. You can get a divergent pathway where the cell does 2 different responses at the same time, right? So we always talk about cells only doing one thing when they're stimulated by something, right? Well, that's not always the case. So for example, epinephrine can bind to the surface of a salivary gland, the alpha and beta receptor, and notice cause 2 dramatic physiologic responses in those cells because they activate 2 different second messengers. You see that? So you get calcium release here by IP3, causes fluid secretion, and then CAMP activation of adenylate cyclase causes the release of the enzyme, amylase. Right? So what do you do when epinephrine binds? You release fluid-rich saliva high in amylase. Right? This is called a divergent pathway because it has 2 different physiologic effects, right? Or 2 different processing effects. Convergent means by binding both, they both converge on the same process and cause triple the effect, right? And so both second messengers, calcium and cyclic AMP here, activate the same enzyme, which causes the breakdown of glycogen. So you release lots of sugar, not just a little bit. And that's called a convergent pathway. All right,